Okay. Uh, I want I want apologize you because my delay. Okay, let's go start. Okay, what was our last session lecture? You know, you remember or no? Can you tell me? You know, Marve? I think you know. Okay, endospore. Thank you. I want loading my file. Please wait. I'm waiting for loading our slide, okay? Okay. Let's go to endospore. No, I think I learning you. Okay. It's a slide. Yes, Marvin. It's a slide. Okay. Endospore. Do you know what is endospore? I think I learned you last session. Do you know and or do you remember what is endospore? Oh, yes, yes. In physio, what I tell you last session. Thank you, Fatima. I call you and want you right. Okay. I have your no sound is okay. Go upstairs. Okay. I want to change my files. Just a minute, please wait. Okay. This is new slide. Yes or no? I think new. Okay. If you are okay, we are start. Fatima Mohammed, this is like you see before or no? Okay. For a study about shaping of microorganisms, we use some method or some section. First, method of laboratory diagnosis. We study about this. Second, methods of bacterial cultivation, identification of bacteria, bacterial metabolism, media for bacterial growth, and sterilization. We should study this. Okay, cut off. That's okay right now or no?
Illustrate key concept of the food. It say
Whatever,
microbiology
this is because of internet. It's
it's
very
a
bad.
big
Here,
field of
here,
microbiology study.
bacterioscopia means microscopic examination. No sound, it's okay. Hey there, Mustafa, do you hear me? Okay. Fatima, please go out class and come back. It should be okay. Microscopic means microscopic examination. Second, bacteriological means method which use culture. Detection sensitivity of bacteria to antibiotic, serological, biological, and DNA texturalities means PCR, polymerase chain reaction. Okay, it is right. I want to last section. slide. Next slide. Please write. Okay. Last session, you studied the different book and different lecture. And now we want to speak about this. They are in your class and lecture. That lecture finish. We start about this. Okay? And the book or no, one lecture in 10 or 10 paper. I told you, I say to you which sentences you should read, you should study. Okay, write this. Methods of laboratory diagnosis: bacteriological, bacteriological, detection sensitivity of bacteria to antibiotics, serological, biological methods, and DNA method or PCR. What this means? Okay, this is culturing. No, this is culture. In the clinical laboratory, it is necessary isolate bacteria in pure culture. What means this? Pure culture. At first, we should can isolate bacteria in pure culture. Bacteria for demonstration their properties such as a study of morphology like this or cultural, biochemical, antigenic and pathogenic properties. Bacteriophage and bacteriocin suspectivity. We should we should we should demonstration all of these in bacteria. And we should determine a sensitivity to antibacterial. Okay, please write and say this is culturing. In this we have only sample, means urine sample maybe. And with this we culture in pure culture. And this in this we study biochemical chemical in bacteria. Please write. Okay, next slide. This is cultivation, means passage bacteria on culture, pure culture. Have some methods. First, we say strict culture or surface plating. This is plate. The include is spreaded thinly over the plate of a culture media in series of parallel lines in different segments of the plate. On incubation, well separate colonies are obtained over the final series of streak. This is a streak culture. Do you understand? Okay, please write. I'm waiting for you for write because maybe I didn't give you this slide. Why? Because I want you have activity in my class. Okay, next slide. What do you say here? Okay, this is method of streak of culture. First, we should painting all of surface in plate like this. Now, second, we can have this for colony. For unique colony, we have this like this. In here we have unique colony or this. There are methods of cultivative. Okay, next slide. Methods of calculating. Lawn or carpet culture. This all of surface of plate we coloring. We have coloring. Yes, Aki, yes. Yes, that's right. Lawn or carpet culture. Lawn cultures are prepared by flooding the surface or plate with suspension of bacteria. It provides uniform surface growth of bacteria. It is useful for bacteriophage typing and antibiotic sensitivity tests. You do it. Please write. In laboratory, you do it before. Okay. Yes, we can. We can, Aki. We can. We can repeat. Okay. When you write, tell me, go to another slide. We continue. I should connect charger. Okay. A slide after this. Okay, before here. Do you mean this? Whatever. This or this? Please write faster, please. دهمین تا 5 املا فقط املا تا 5. Okay, right. Can I continue or no? Fatima Agri, can I continue or no? Okay. Another method for cultivation we call extract culture. It is made in tubes, not petri dish, containing agar slabs. It is used for providing a pure growth of bacteria for a slab aggregation, maybe. It is right. Do you have question? Please ask me. Only for mention your attention. This is Italy. It is an important. Yes, this is third method for calculating. When you write, please say me continue. Okay. Another method is step culture. It is prepared by punctuating with charge line straight wire, means loop. A stab culture is employed mainly for cultivation of antibiotic. Here in this, here we have an odor and anaerobic bacteria can grow here. Okay, you understand? Okay, this is plate culture. When we want to make pure plate culture, we use this. Here, please look here, please look here. We have original sample or original inclusion. We use dilation in each tube until here. For example, here is 1 to 10, here is 1 to 100,000. It's very, very, very more. Here in culture, we have a lot of colony in pink, but here we have only 1 or 2 or 4 colony. You know? You understand? Okay. Another method we called liquid culture. Here we have liquid. Previous method is solid with agarose. Yes, agarose gel. Okay. In liquid culture, in a tube, bottle, or flask, one of them may be included by touching with charge loop. We charge loop and have culture on this. This is liquid culture. We transfer with loop our specimen here. This is okay. I will explain for you. Zayed, Zayd, Razza. Please write this slide and you understand or not? Okay. Okay, I'll explain for you. Charge means you take a specimen with loop. Charge. You know, Abdul Qudus, first time we take spikes with loop and we repeat it. It means charge. Okay. Your friend wants me explain this slide more. This slide. Look me, we have 5 tubes and 5 petri dish. Our petri dish is pure, but our tube are not. We can culturing our sample, our specimen or sample here. This is first tube. In our first tube, adulation is 1 to 10. But in last tube, adulation is 1 to 100. It means more dilution and less colony we can have. If we have culturing from first tube, we have this culture. All of bacteria like furniture in our plate. But in the last culture, we have only 1 or 2 or 5 colonies. Because a lot of colony loss. You know? Do you understand? Do you understand? Okay. Next slide. Identification of bacteria. Microscopic examination. It helps to detect a shape, a size, and arrangement of microorganisms. A staining reaction. On gram staining, we can have 2 groups of microorganisms, gram positive and gram negative. It is very, very easy. Please write. Please write. When you write, please tell me. This is gram. What is this? Gram A. This is gram negative. Yes, Fatima. Yes. Marhaba. Gram negative means what? From the culture, from the timing. It is either red or pink. And B is gram positive because it is purple. Okay, it is right. When right, please call me. I continue. Okay. Identification of bacteria from what? First, for now, we are knowing moving, for example. Motility. Some bacteria can move, like Salmonella, E. coli, Pertussis, Pseudomonas, Vibrio, Clostridium. But there, in dark ground microscopy and fast controls for studying multiplicity is very hard. And we can or we should, especially stain for algae. Like here, okay, please write and tell me. Physiology? Yes, this is physiology. And this physiology is necessary for moving. Bacteria moving by this physiology. Okay, let's go next slide. Identification of bacteria. Culture character. Growth, recruitment, colonial characteristics in culture. Means, for example, form. Like this, or circular, or fragments, or irregular, or residue, or spiral. Can have this form in culture or if we want to speak about elevation of colony, can be flat or raised or convex or okay. I can't do anything more because this is because of net, internet. About margin, margin means here. It can be entire, undulate, filamentous, lobate, arose, or cured. Please write this, know this, don't know. Please like this and this. I'm here. Yes, this is important. Okay. If you write, please tell me. I continue. No, rest, no. Rest half past 4 because we have class. Okay, I'm here. You can write it. When you write, please call me. Please tell me. Continue. Excuse me, doctor. Will you send us? No, no, I didn't. I didn't send you. Maybe I sent, but no. Please write. Please write, Haidar. Maybe I will send you. But I want, by writing, you be active in my class. Okay? Why you don't understand, Fatima? Answer me and I will explain you more. Why you don't understand? I'm here for explaining you. Yes, I will send you, but right and here, please answer me everything you don't know. Okay, I explain you more. It is my duty. Okay, what you don't know, what you don't understand, please tell me. I will explain for you. Okay, let's continue. This is colony morphology. Look here, look here. Colony morphology. I called you. It isn't very important. Please only listen in class. Okay, this is colony. Rough colony, red colony. Okay, okay. Identification of bacteria. Metabolism. Capacity to form pigment and power of hemophilus in help for classification of bacteria. Like this, we have two classification of hemolysis in bacteria. One is alpha, another is beta, another is gamma in red or blood agar. Look here, please write this and look, I will continue to you. Yes, I want send you slides, okay? Please wait. I send you a slide in your class. Okay? Only because of Fatima. Fatima Adnan, only for you. But please, here writing, because by writing you remember more and you understand more. Okay, let's go. Fatima, you are one of my good students. I see your attention in class. Okay, let's go. Let's go. Another slide. You know, this is colonies pigment. We have colony in different color. Here, blue, red, yellow, green. You know, it's very beautiful. No, okay. Please, is biochemical reaction. The more important and widely used tests are under fourth sugar termination. Here, yes, Fatima. There are three different bacteria and three sugar fermentation. The three sugar fermentation. One is this: no sugar fermentation, no acid, no glass. Some bacteria no without any of these functions. Another have acid but no gas, and this have acid and gas and fermentation. Acid and gas is product of fermentation, sugar fermentation. Do you understand? Okay. Let's go. Another method for identification of bacteria, which is indole production or hydrogen sulfide production. Like here, we have indole positive in this bacteria. And here, sorry about my coughing, we have hydrogen sulfide production here. Okay, this line, thank you, Fatima. It is allergic. Okay, for identification of bacteria, we have another test. For example, citratolization or nitrat radiation or methyl red test or orysis test or catalyst test or oxidized oxygen. This is oxidized, catalase. Staphylococcus have catalase. They can catalase H2O2 to H2 and O2. This is catalase. No? Or they can, but Staphylococcus can't catalase. They haven't catalase. This is one test for identification Staphylococcus by strep. Do you understand? Have next slide. Here, here. This is biomerox or strip test. This. There are strips tests. 20 tests are performed on this strip. One strip, two strip, or three. 20, 1, 2, 3, 4, 5, 6. 20 strips by a simple produce, saving time and money. For example, here for termination, here for indole, here for example, acid, here for gas. Okay, and in different bacteria we use in this step. For example, Escherichia coli, Enterobacter agglomerans, Edwardsella hoshi. Okay, have different reaction in different bacteria to this simple. You understand? Okay, let's go. Yes, this side. Here, here, identification of bacteria. Antigenic analysis. By using a special sera, we can identify microorganisms by coagulation reaction. We call this serologic type of reaction. For example, for serologic typing of Shigella, we use this. In this picture, please here, the climbing of bacteria is seen in this circle. Bacteria climb. Okay? But in this circle, we haven't any climbing of bacteria. Okay? Do you understand? Let's go next slide. Okay, let's next slide. Do you hear me? Okay. Here we have a bacteriophage typing. Phage bring about disease of susceptible bacteria cells. For example, here in E. coli we have this light of colony, or E. coli A we have this, E. coli K, E. coli orange. Okay. Another identification method is pathogenicity. For pathogenicity tests, commonly used laboratory animal models are quinoa pig, rabbit, rat, or mouse. Their resistance to antibiotics and other agents. For example, here, this is resistance, but this is sensitivity. This is what is this? What is this? Do you know what is this in plate? Yes, there are different antibiotics. Okay. Metabolism is the process of building up chemical compounds in the cell and then breaking down during activity to receive the required energy and the building element. Metabolism compares of anabolism and catabolism in bacteria. Do you understand? It is very easy. Do you understand? Do you have any question? Another method is chemically or bacterial constants. Okay, waters 75 or 85% brown water and free water here. Or dry matter 50 or 25 organic parts of and mineral substance in organic parts like this. In bacteria, we haven't any water. Okay, we haven't any water. Okay, Marve, please write this slide for next section. We should continue from slide 26. That's enough for today. We have microbiology with another student. Please go and have rest We start on 15 minutes ago. We will start. Okay, do you have any question? Please ask me. If you have any question, please ask me. No, they didn't come for exam. Teacher didn't. Okay. I will see you in next class, microbiology. Thanks a lot.